Hey everyone, this is Dr. Marcon starting the lecture series on the cardiovascular system and cardiovascular physiology. First, we're going to look at a little bit of the anatomy of the heart. We'll talk about, we've already talked about the three main components of the cardiovascular system, the three main components being the heart, blood vessels, as well as blood itself. And we talked about blood in the last chapter. So the primary function of the cardiovascular system is to deliver nutrients and remove waste. Humans are said to have a closed cardiovascular system because all the vessels are interconnected with one another, and down to the capillaries where you have sites of exchange, either respiratory gases, nutrients, etc. Arthropods or mollusks are said to have an open cardiovascular system, meaning whatever fluids are pumped through the cardiovascular system then goes out and surrounds the organs and becomes interstitial fluid. The circulatory system is actually the functional name of the cardiovascular system. So blood will circulate in humans in a closed system, again meaning it's all interconnected down to the capillaries where and these are the sites of exchange. And again, in an open circulatory system, fluids will bathe organs and structures within the organism. So here we see a grasshopper. Its fluids circulate to the heart, and then as hemolymph goes into sinuses surrounding organs and becomes interstitial fluid. And then another example of a closed system, we see this earthworm. So the earthworm, it has a closed circulatory system, meaning its fluids do not go out into the interstitium or out to the tissues to become interstitial fluid. Rather, it circulates in this closed system, going down to the smaller branched vessels in each organ. Now we'll talk a little bit about the anatomy of the heart. We know that the heart has 4 chambers. The 2 upper chambers are known as the atria being the plural form. We have a right atrium and a left atrium. The atria are the receiving chambers, receiving blood on the right either from the system or on the left receiving blood from the lungs. These atria are very thin-walled, and they pump nearby to the ventricles or downward towards the ventricles. So here we see the right atrium on the right receiving blood from 3 major vessels, being the superior vena cava, the inferior vena cava, and the coronary sinus, which drains deoxygenated blood from the heart. And then on the left we have the left atrium with that little outpouching on the both right and left side. It's called the auricle, just basically providing more space for blood to enter into the atrium. The next 2 chambers are the 2 lower chambers. These are the ventricles. So we have a right ventricle. The right ventricle will pump blood to the lungs, and this is deoxygenated blood. And then we have the left ventricle, which pumps oxygenated blood coming from the lungs basically to the rest of the body. And we can see grossly in a gross specimen of the heart that the left ventricle has a thicker wall because it has to actually pump blood farther going to the rest of the body. So if we have these lines that kind of separate the 4 chambers, we can see the different septums that separate the 4 chambers of the heart. Between the right and left atria is the interatrial septum, that wall dividing the right atrium from the left atrium. And then we have this atrioventricular septum, which will be important later when we talk about the conducting system of the heart. So this atrioventricular septum separating the right and left atria from the right and left ventricle. And then we have this very thick-walled interventricular septum separating the right ventricle from the left ventricle. And if we just do a kind of basic schematic of the four chambers of the heart, just a basic Valentine's heart. As you can see, we have different walls separating the four chambers. So we have the right atrium, the left atrium separated by a wall, and then the atria separated from the ventricles, and then we have the right ventricle and the left ventricle and a wall separating the two ventricles. This slide shows the flow of blood through the heart. The blue arrows represent deoxygenated blood returning from the rest of the body to the right side of the heart. Now, deoxygenated blood is actually a bit of a misnomer because there are still oxygen attached to the hemoglobin within these red blood cells. However, because the blood has already flowed through the tissues, it is only 70% saturated with oxygen. So, meaning for every 10 hemoglobin sites, there are only 7 that are attached to oxygen. Now, it is blue because there is less oxidation of the hemoglobin. So, less oxidation means less red color, and therefore it has a distinctive blue color because there is less oxygen. Now, all the deoxygenated blood returns to the right atrium. So we see here blood returning via the superior vena cava as well as the inferior vena cava. We also have blood returning from the actual blood supply of the heart via the coronary sinus posteriorly. So all deoxygenated blood returning to the right atrium. Now, about 2/3 of this blood will flow passively into the right ventricle. So the majority of the blood flows passively into the right ventricle. 1/3 of it is actively pumped during depolarization or contraction of the atria. So I'm just going to talk about the flow of blood through the heart right now. So from the right atrium, blood will then pass through the right atrioventricular valve, also known as the tricuspid valve. It has 3 cusps, so we call it the tricuspid valve. Blood will then enter the right ventricle. Now, during ventricular depolarization or ventricular contraction, blood will flow through the pulmonary semilunar valve. They're called semilunar valves because if you dissect them out, they look like half moons. So they're half moon in shape. So through the pulmonary semilunar valve or pulmonic valve, blood will then flow up the pulmonary trunk, entering the pulmonary arteries right and left to travel to the lungs, where blood will then become reoxygenated. So blood within the lungs becomes about 99% saturated with oxygen. This oxygenated blood will then travel from the lungs through the pulmonary veins, right and left, to enter the left atrium. So here we see blood returning back to the left side of the heart via the pulmonary veins. Reoxygenated blood will then enter the left atrium. Again, this is just another atrioventricular valve where blood will pass. This is the bicuspid or mitral valve. So from the left atrium through the bicuspid or mitral valve, blood will enter the left ventricle. From the left ventricle, blood is then pumped through the aortic semilunar valve or aortic valve to go to the rest of the body. We can see that the part of the aortic valve as well as the ascending aorta is actually posterior compared to the pulmonary trunk here. So behind or posterior to the pulmonary trunk is the aortic valve and the extending aorta. Blood will then travel to the rest of the body. We talk about the heart undergoing a contraction relaxation cycle to allow for blood to be pumped to various areas. Now, the systolic phase allows for blood to be pumped out. This causes the highest amount of pressure within the chambers. The diastolic phase allows for chambers to fill, where we'll see the lowest amount of pressure in the chambers. So the pressure in these chambers decreases, allowing for filling of these chambers. Now, when the atria contract, the ventricles will relax. This is known as ventricular diastole. And then when the ventricles contract, the atria will relax, and this is known as ventricular systole or systole, depending on however you want to pronounce it. So contraction is known as systole. Blood, if systole is occurring within the ventricles, blood is pumped out either on the right side towards the lungs or on the left side towards the rest of the body. And this is where we have our highest pressures. If there's contraction within the atria, the atrial pressure will increase, allowing for blood to be pumped into the ventricles. So as blood is pumped into the ventricles, the pressure within the ventricles will decrease, allowing a filling of blood within the ventricles. So contraction is synonymous with depolarization. So depolarization first occurring within the atria, and then a wave of depolarization traveling down into the ventricles, but we'll get into the electrical conductivity in just a little bit. Blood flows from high to low pressure, and the pressure differences will dictate the flow of blood. So with contraction of a chamber, this increases the pressure. So the pressure pushes the blood into the next chamber, or pushes the blood either to the lungs or to the rest of the body. So if we have an increase in pressure, for example, in the right atrium, we'll have a low pressure or decreased pressure within the right ventricle, allowing for the right ventricle to fill with blood. So again, these pressure differences allow for the flow of blood from one chamber to the next, or either to the lungs or to the rest of the body. So again, the pressure in the ventricles has to be higher than the pressure in the atrium for blood to flow during systole. So as the ventricles contract, this pressure increases, whereas the pressure within the atria decreases, allowing for blood to fill within the atria before another cycle occurs again. We said earlier that the majority of the blood that enters the atria will flow into the ventricles passively. So about 2/3 of this blood will flow from the atria to the ventricle passively, whereas 1/3 of the blood within the atria are actively pumped into the ventricles. The numbers that you see in this picture represent systolic pressure over diastolic pressure. So the pressures during contraction and relaxation respectively. So for example, in the right atrium, during the systolic phase in the right atrium, the pressure in the right atrium equals 5 millimeters mercury. And during the systolic phase of the right atrium, the right ventricle is in diastole, or ventricular relaxation, and this pressure is at 0. So this pressure difference, the pressure being higher in the atrium versus the ventricle, will allow for this active flow of blood from the right atrium into the ventricle. So this pressure pushes the blood from the right atria into the right ventricle. Now, during systole within the right ventricle, the pressure within the right ventricle rises to 15. So the pressure will push the blood from the right ventricle through the pulmonic valve, through the pulmonary semilunar valve, up the pulmonary trunk, through the right and left pulmonary arteries, into the lungs. So there's a large increase of pressure in the right ventricle during systole, whereas the pressure within the right atrium decreases to 0 to allow for filling of the right atrium with blood. So again, it's this pressure difference that allows blood to flow from a high to low pressure within the chambers. When we talk about blood pressure, what we're really actually talking about is aortic pressure, or pressure within the aorta. Notice that the systolic pressure within the aorta is just the same as the systolic pressure in the left ventricle, because basically the left ventricle is the structure just before hitting the aorta. So systolic pressure we say is representing aortic pressure, and this is the pressure that the aorta is using to force blood to the rest of the body. And its diastolic pressure, or its filling pressure, is 80 or 80 millimeters per mercury. Notice that there is a difference between diastolic pressure within the aorta as well as diastolic pressure within the left ventricle. Now, diastolic pressure in the left ventricle has to go back down to 0 in order for filling to occur, because this pressure has to be below whatever the systolic pressure in the left atria is. So we know that the left atria, its pressure is 5 during systole, or during contraction of the left atria, whereas ventricular pressure on the left will be 0, allowing for blood to flow from the left atria to the left ventricle. And then once the left ventricle undergoes systole or systolic, the systolic portion of its cycle, it will then push blood into the aorta, and the filling pressure of the aorta is 80, which is of course less than 120. So again, this pressure difference allowing for blood to flow from the left ventricle into the aorta. Now, aortic pressure has to be maintained at 80. Why is that? It's because the elasticity of the aorta, we know that most great vessels have that middle muscular layer that also contains elastic connective tissue. It's this elastic tissue that allows for the pressure within the artery, within the aorta, to be maintained at 80, allowing for blood to flow to the rest of the body. Now for venous pressure. Venous pressure, we saw in lab, can be measured between 4 to 10 millimeters mercury. So the systemic vein should have a pressure higher than 5, so 5 being the pressure in the right atrium, and to allow for blood to flow back to the right atria. Venous pressure actually represents continuous flow from the tissues, so it will be only represented by one number, and this number is anywhere between 4 to 10 millimeters mercury. So this pressure has to be higher in order for blood to flow into the right atrium. And during its relaxation, the right atrial pressure goes down to 0, so that blood can flow from the veins with a higher pressure between 4 to 10 into the right atrium. So again, this venous pressure has to be higher than the pressure in the right atrium in order for blood to flow from the veins back to the right atrium. This slide just shows how oxygen is exchanged within the capillaries. When blood leaves the left side of the heart, specifically the left ventricle, to go into the aorta, blood at this point is about 99% saturated. So from the left ventricle, blood will flow through the aorta to the rest of the body, and we see different capillary networks throughout the body, within the head and brain, the arms, the trunk, the digestive tract, the liver, down to the lower limbs. So when blood leaves the lungs to go to the tissue, it is 99% saturated. However, once we have gas exchange within the capillary networks, the blood becomes less saturated. We like to say deoxygenated, but though it still does contain some oxygen. So at this point, once there is exchange within the capillary system, within the tissues, blood then becomes desaturated down to about 70%. So at this point, blood is only 70% saturated with oxygen, again giving it that bluish color, bluish tinge to the blood, and then returns back to the right side of the heart. So we have specialized structures within the heart that ensure one-way flow of the blood, and these are our heart valves. The valves within the heart prevents backflow, or what we call regurgitation of blood. We have the semilunar valves. The semilunar valves are shaped like half moons. This structure fills with blood to help close the valves, close the semilunar valves to help prevent regurgitation back into the ventricles. The cusps of the atrioventricular valves, both the tricuspid and the bicuspid valve, they are attached to structures called chordae tendineae, which are of course attached to papillary muscles. So when we have contraction of the ventricles, the papillary muscles will pull on the chordae tendineae to close the atrioventricular valves so that we don't have a regurgitation of blood from the ventricles back into the atria. So these are all the specialized structures that ensure that blood flows one way. So here is just a superior view of a transverse section of the heart. So if we take away the two atria on the superior portion of the heart, we can actually see the different valves that make up the heart. So anteriorly, most anterior, we have the pulmonary semilunar valve, or the pulmonic valve, before it enters the pulmonary trunk. And then just posterior to that, we'll see the aortic semilunar valve open. So if it is open, this means that the heart or the ventricles are in the systolic phase, pumping blood through the semilunar valves. And during systole, the two atrioventricular valves are closed. So we have the tricuspid valve on the right, also known as the right atrioventricular valve, as well as the left atrioventricular valve, or the mitral bicuspid valve on the left. Now, the mitral valve is also known as the bicuspid valve because it has two cusps that make up this valve. The way that this valve was named is based off the hat of the Pope. The Pope's hat is called the mitre, and the mitre has two sides. So, like the Pope's hat, the mitral valve has two cusps. Now, the bicuspid valve is actually more resilient to regurgitation or backflow of blood compared to the three cusps of the other valves. Why is that? Well, because the left ventricle, of course, has more pressure, which allows for blood to be pushed out into the system because it has a longer way to go. So more pressure is needed to push the blood out of the left ventricle. A two-cusp valve is more resilient for the blood to be regurgitated back into the atrium. The heart is composed primarily of cardiac muscle, and if we remember our histology, cardiac muscle is a type of muscle that is striated and has structures that allow for communication between cardiac muscle cells. So the heart is made up of 99% cardiac muscle and 1% of the stuff that holds together. That is the cardiac skeleton that makes up the valves of the heart. And just by seeing this slide, we can see that the wall of the left ventricle is a lot thicker compared to the wall of the right ventricles, because the left ventricle has to push the blood a lot further, so it is a lot more muscular, allowing for that increase in pressure to push the blood from the left ventricle through the aortic valve, through the aorta to the rest of the body. Cardiac muscle is connected by gap junctions, and these gap junctions are important for communication between cardiac muscle cells. So the mode of communication is just a direct transmission of electrical signals between cells via the gap junctions. This allows for the passage of ions, and again allows for that direct transmission of electrical signals. Cardiac muscle cells work in what's called a syncytium. So syncytium, basically they work together through this network of interconnected cells. There are structures in cardiac muscle cells called intercalated discs. These discs contain gap junctions or gap junction proteins, which allow for the coordination of movement and the transfer of electrical signals during systole, during contraction, or diastole, during relaxation. So here we can see a cross-section through an intercalated disc between cardiac muscle cells and cardiac muscle fibers. We also have walls, or septums. Whenever you see the word septum, think wall. So the septums separate the heart chambers. So septums are tissue that divides the heart chambers. There are 3 very important septums or septa within the heart. The most important septum that we want to talk about is the atrioventricular septum. So the atrioventricular septum separates the atria and the ventricles and does not allow electrical signals to pass between the atria and the ventricles. Now, this is important in order to allow for the rhythmic contraction of the heart in a specific way. So the atrioventricular septum, again, that wall separating the 2 atria, right and left, from the 2 ventricles, right and left. Other important septa include the interatrial septum. This is the wall that divides the right atrium from the left atrium, and we know that during fetal life there was a communication between the 2 atria called the foramen ovale, foramen being a fancy word for hole. So there was a hole allowing for communication between the right and left atria, which eventually, after reaching adulthood, or after the baby is born, the hole closes and becomes the fossa ovalis, which we can see in models as well as gross specimens of the heart. The next wall that we want to concern ourselves with is the interventricular septum. This is the wall between the 2 ventricles. Now, what's important is that electrical signals can pass through these 2 walls between the interatrial septum as well as the interventricular septum, allowing for coordinated either contraction or relaxation between the 2 chambers. So electrical signals can travel within the atria and within the ventricles, but not between. All these contractions and relaxations are coordinated due to specific cells, and these are autorhythmic cells that can spontaneously generate their own action potentials. Within the heart, we have nodal tissue, so we have different cardiac nodes within the heart that will generate the action potentials that allow for contraction or relaxation of cardiac muscle tissue. Now, the GI also does have autorhythmic cells within the smooth muscle, and basically, we're not going to get too much into it, but these vasoelectrical rhythms within the smooth muscle allow for peristalsis, or the movement of either fluids or food within the digestive system. So within the cardiac system, or within the heart, we have our cardiac nodes. We have the sinoatrial node, which is located in the superior portion of the right atrium, just where the superior vena cava meets the right atrium. And then we have the atrioventricular node at the base of the right atrium. And then within the ventricles also have their own ventricular nodes dispersed out within the ventricles. Here on this slide, we can actually see potassium ions moving out of these cells. Now, the action potentials within the heart solely depend on movement of sodium ions as well as potassium ions. So we'll get into how action potentials are generated. Basically, we have action potentials occurring first with we have our set resting membrane potential, then we have depolarization, usually depolarization occurring due to an influx of sodium. And then depolarization, we then have initial repolarization. So sodium channels begin to close, and then the cell begins to repolarize as potassium will leave through open potassium channels. But we'll get into that in just a little bit. First, we're going to talk about how electrical signals travel through the heart. We know that the sinoatrial node is known as the pacemaker of the heart because it sets the rhythm of the heart. It's what first generates that electrical impulse. So a wave of depolarization starts from the atria to push blood from the atria to the ventricles. And we know that electrical signals are passed via gap junctions between cardiocytes or cardiac cells. And they help coordinate contraction. So again, a wave of depolarization starts from the atria, starting with the SA node. Oops, let's go back. Starting with the SA node. And then the signal will then travel after it has dispersed out throughout the atria. It will then travel to the atrioventricular node. It's important that the signal stops at the atrioventricular node because we want coordinated contraction within the ventricles. So from the atria, the electrical signal will pass to the AV node, and once it hits the AV node, the electrical signal will then travel down the interventricular septum. So depolarization travels down the interventricular septum to the AV bundles, so to the right and left bundle branches, and then to the Purkinje fibers. So here we can see this wave of depolarization starting with the purple here at the SA node, traveling down throughout the right and left atria, then hitting the atrioventricular node, to the AV bundles, to the right and left AV bundles here within the interventricular septum, then going down to the Purkinje fibers and allowing this wave of depolarization to go up through to the ventricles. So this allows for depolarization to occur from the bottom of the ventricles to pump up, allowing for blood to pump from the ventricles and then through the semilunar valves. We want that action to occur because we don't want ventricular contraction occurring at the top part of the ventricles. We want the ventricles to contract from the bottom so that blood is pumped up to either the pulmonary trunk or the aorta. Think of trying to take toothpaste out of a tube. We don't want to, push the toothpaste from the middle of the tube. We want to push the toothpaste from the bottom of the tube. So we want depolarization to start at the bottom of the ventricles to allow blood to be pushed up into either the pulmonary trunk or the aorta. So cardiac nodes have different inherent rates. We know that the fastest node will set the speed for all cardiac muscles, and the node with the fastest speed is the sinoatrial node. So the SA node contracts at about 70 to 100 beats per minute. So this is our inherent heart rate. This is our normal heart rate. The next fastest is the atrioventricular node at 40 to 50 beats per minute, and then the ventricular nodes at 20 to 30 beats per minute. So the SA node sets the rhythm. The atrioventricular and ventricular nodes are deactivated by the SA node by setting them farther away from threshold through hyperpolarization. So the fastest node will inhibit the slower node via overdrive suppression. Again, the SA node will hyperpolarize the AV and ventricular nodes. This is the trait of the fastest node, basically putting them farther away from their threshold. Now, if the SA node becomes damaged, the AV node would then become the fastest node and set the rhythm. And then the heart rate would go from 70 to 100 beats per minute to 40 to 50 beats per minute. If the AV node is damaged, then the ventricular nodes would take over, and our heartbeat would then go to 20 to 30 beats per minute. So basically, multiple nodes create sort of a backup system within the heart. I mean, would we be able to live on 20 to 30 beats per minute? I mean, we'd live, but it's not ideal. It does not create an ideal situation. Ventricles, of course, can be impatient. We have what's called premature ventricular contractions, or PVCs. This is when the ventricle beats early. When this happens, the fill time is shortened and the volume that is pumped by the ventricles decreases or becomes lowered. And usually this can be due to reduced SA node firing or a lack of overdrive suppression, a lack of that hyperpolarization occurring or due to the SA node. Now, this can be done due to the intake of certain stimulants such as caffeine or cocaine. Also, a lowered heart rate. Usually athletes are able to decrease their heart rate because they're able to pump their blood more efficiently. However, this can also cause premature ventricular contractions, or PVCs. Heart range can vary, can range widely between individuals. A heart rate below 60 beats per minute is called bradycardia. Bradycardia can be due to a number of reasons. It can be genetic. For example, you could just have a slow SA node. Or you can have an active parasympathetic nervous system. So you know that the parasympathetic nervous system is responsible for our rest and digest functions of the body. Exercise can increase stroke volume, and we'll talk about stroke volume and cardiac output and how it relates to heart rates. Also, disease, any pathological conditions can cause bradycardia. We consider a heart rate between 60 to 100 to be normal, so that when we take a person's pulse or heart rate in a clinical setting, ideally we want their heart rate to be between 60 to 100 beats per minute. And anything above 100 is considered tachycardia. So brady meaning slow, tachy meaning fast, so above 100 is considered tachycardia. And there are all sorts of causes that can cause tachycardia, being, sympathetic responses, exercise, when we get our heart flowing by running on an elliptical for 20-25 minutes, which is what I'm doing now, considering we have no other real means of exercise, staying at home. So just a quick recap. We know that the sinoatrial node sets the basic heart pattern. The SA node spontaneously depolarizes, has that autorhythmicity characteristic to it, and anywhere between 70 to 100 beats per minute. So the SA node spontaneously depolarizes, again allowing for contraction. This wave of depolarization will then spread through the atria and then hit the AV node. I think of the AV node sort of as like a toll where all cars will kind of descend upon and stop for just a few seconds, at which point the electrical signal will then, or the depolarization will slow, the conduction will slow through the AV node, and then depolarization will then travel from the AV node through the bundle of His. From the bundle of His within the interventricular septum, it will then travel to the right and left bundle branches towards the apex of the heart. The depolarization will then spread upward from the apex to the Purkinje fibers, and this, of course, allowing for ventricular contraction starting at the bottom of the ventricles, allowing for blood to be pushed up the ventricles, going either towards the lungs via the pulmonary valve and the pulmonary trunk, or to the rest of the body via the aortic valve and the aorta. So that's about it for now. This will be part 1 of your cardiovascular physiology. We will get into more of the conduction system and EKGs in the next video.